Okay, good afternoon, everyone. It's my great pleasure to welcome our guest today. First of all, I'd like to introduce myself. I'm Attila Gácser. I am a mentor of this program and from Szeged University. And I'm a host of our today's speaker. And this is an honor to welcome in Szeged Professor Dr. Joshua Nosanchuk from the Albert Einstein College of Medicine, New York, USA. And I would like to introduce him in a couple of sentences. So Dr. Nosanchuk is a professor of medicine in infectious diseases, professor of microbiology and immunology, a senior attending physician, and senior associate dean at Albert Einstein College of Medicine. He is also an advisory researcher for the Center for Technological Development in Health for the Oswaldo Cruz Foundation of the Brazilian Ministry of Health. His laboratory focuses on pathogenic fungi, including neglected organisms, and the development of novel therapeutics. He has published over 325 scientific papers with over 21,000 citations and written 20 book chapters. Dr. Nosanchuk is one elected fellow of the American Society for Clinical Investigation, American Academy of Microbiology, American College of Physicians, and Infectious Diseases Society of America. He has had wide-ranging leadership activities in medical societies, including the U.S. National Board of Medical Examiners and Infectious Diseases Society of America. He is one of the editorial boards of several journals, including the Journal of Infectious Diseases and PLOS Neglected Diseases. Professor Nosanchuk has a long-time collaboration with the University of Szeged. He trained several PhD and postdoctoral students in his laboratory from Szeged, published over 40 papers, and supported numerous grants with scientists from the University of Szeged. And with this, I would like to welcome you, Josh, and please hold your talk.
So it's really a pleasure to be here today, and I look forward to sharing some information with you. I didn't know I was going to give this talk until about 3 weeks ago when Attila asked me to talk to you, and he asked me to give a talk about general mycology as well as tie it into some research and some infectious diseases. And it was very timely because of the WHO putting out a fungal pathogen list. So I put this talk together over last weekend, and I hope that you will enjoy it because it is very broad and I think touches on some very interesting aspects of the fields that some of us have invested a lot of time in. So we'll take off here. When I give a lecture about certain things, people are really interested in it. They think, oh my God, that's so cool, methamphetamine. You know, all these people are dying and doing these drugs. But when I give a talk about fungi, people kind of move away from you if you tell them at a bar that you're working on fungi. So I want you to understand that fungi are incredibly important. So we're going to learn about why we should care as we go through this. And it's amazing because prior to the 70s, prior to the 1950s, they weren't very common and we didn't really study them very much. But if you look at the burden of disease, there are more than 6 billion people who have significant fungal diseases, with about a third of those being very significant. It causes at least 1.5 million deaths a year. That's a lot of deaths. And in terms of costs, in the US, it was calculated that it's almost $7 billion in medical costs. And many of the organisms that we work on kill over 40% of the people that have a significant infection with them. So really important. Just some basic microbial comparisons to make sure we're all on the same page. The biggest thing to remember is that they're eukaryotes. And since they're eukaryotes, it means they're very similar to us, which makes combating them very hard, because if we create a drug, many of those drugs will also adversely affect us. So they're eukaryotic. They do have some things that are different in the cell wall, chitin and polysaccharides, and chitin is a structure that we're trying to target with some of the drugs that are being developed. They're amazing because without fungi, all the leaves, all the trees, all sorts of stuff would just accumulate. The fungi are really the organisms that break all of that down. So this is the only work I'm going to ask you to do. How many fungi are there? How many are human pathogens? And how many do people like me as infectious diseases see them very often? I'm not going to make you say it out loud. We're just going to go on, but think about it. You thought about it? Good. So there are at least 1.5 million. Some people say there are over 5 million, but there are at least 1.5 that have kind of been categorized. And about 300 are human pathogens. And I see about 20 different species a year in my clinical practice. And this also shows how amazingly diverse they are in terms of seeing things like, here we go. This is Candida albicans with long hyphae and these budding yeast off of them. Histoplasma with all these cells within a macrophage. This is a mushroom called the honey mushroom, which can spread more than 3.5 miles, so over 5 kilometers. So tiny, tiny to very, very large. Amazing diversity. And again, they do so many different things. They're incredibly important in making wine, which we had some great Hungarian wines at lunch today. But they also can kill a lot of vegetables and plants. They can cause cancer. And the bottom part is really important for this talk in terms of killing mammals, insects. So they're all about us. Every breath you're taking in, you're inhaling some spores. There are fungal spores in the air. And depending on where you are, there are a lot more. So if you're in a tropical rainforest, there's a lot more than if you're in Antarctica. But we don't get sick very often because usually our host immune response is very effective at getting rid of it. And again, in the pre-70s, pre-50s, we didn't see much fungi. People didn't live as long. We didn't put people in ICUs, intensive care units, and hospitals. But over the past several decades, we've done a lot of different things. And climate change is really important. And I'll go through some of that. Global travel. I was across the world yesterday, and it's so easy to go places. So movement of us, movement of goods, spreads a lot of diseases. And our patients are changing tremendously. The thermal barrier is very, very important. Only a tiny amount of the fungi out there can survive at high temperatures. So sometimes when you have a fever, people want to give you drugs to make the fever come down. Fever is actually very good. It kills a lot of the infecting organisms that come into us. So if you can get above 37 degrees, you can cause disease in us. If you can't, you can certainly kill trees, you can kill insects with the zombie ants, and we've had large die-offs of some of our amphibians, like the chytrids that have died off in places across the globe. So this is really important for you to think about. Why are these organisms that are primarily environmental? Most fungi are not human commensals. Candida albicans, Pneumocystis, Jerevecii, those are some oddballs that really only live in us. But most are out in the environment. Why are they causing disease in us? There's a constant battle going on in the environment with these organisms and worms, with these organisms and amoeba. And these are examples of some different fungi interacting with macrophages on the top here on the left, as well as amoeba on the bottom on the left. And these are all amoeba on the other side, showing that these organisms are taken up into phagosomes. And they can replicate inside of those phagosomes. Sometimes they kill the amoeba, sometimes they kill the C. elegans, sometimes they don't. So there's a constant battle going out there that's been happening for millennia, which is why some of these organisms are so effective at causing disease in us, because some of the same techniques that have been developed in these boot camps in the environment are able to then subvert our host responses and cause disease in us. So climate change is really important. COVID-19 is a great example of changing environments and travel and the hosts. Certain people get much sicker. So lots of things change with COVID-19. I'm going to give you one example. This was me, and this was the pandemic. So lots of things change. Lots of things change. Just sharing a little bit. So if we look at the past 100+ years in infectious diseases, there have been a lot of milestones. Sanitation was really the biggest change for protecting human health. When you're not living in filth, you're not getting as sick because that burden of bacteria and viruses and other organisms are separated from you. And then, of course, we had serum therapy, and serum therapy has come back. Monoclonal antibodies and convalescent serum. We all see and hear about that in the news for COVID-19 and Ebola and other diseases. And then we had vaccines, and then we had penicillin. And we saw a remarkable dive in infectious diseases. In fact, in the 70s in the United States, the Surgeon General, the head of medicine in the United States, said the war against infectious diseases is done. And places like New York City, we started taking apart some of our health systems for infectious diseases, which was not a good idea because right after that we had the HIV pandemic and we had tuberculosis shoot up because we had lost our infrastructure to control public health. And along with the HIV pandemic, we also started doing much more in terms of organ transplants and giving chemotherapy and putting people in intensive care units and putting catheters in and doing more hemodialysis. So changing our host tremendously. And global travel, of course, really shot up starting in the late 70s and into the 80s. And along with that, we started seeing drug-resistant organisms. Fungi started coming up. And really, opportunistic pathogens is a major theme in terms of what we see in our hospitals today. And it's really about a change in our hosts. And it's really important to think about hosts because certain organisms really only cause disease when you have a weakened immunity. So Candida albicans on the top left there is a classic example of something that lives with us all the time, and we don't get disease unless there's disruption. If you give antibiotics, women are very susceptible to vaginal yeast infections. If we knock out someone's neutrophils with chemotherapy and we put a catheter in their arm and we disrupt all these barriers, invasive candidiasis is very common. This is a list of various biologics that are now being given to our patients. It's really long and it's growing, and it knocks out all sorts of parts of the immune system to prevent diseases, whether it's something like an immunodeficiency or whether it's an immunoinflammation disease. All of these have unintended consequences of linking to a fungal infection. And you see Candida, 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 but there are also things like histo and blasto that come up in these patients too. So the more we do in terms of advancing medicine, the more challenged we get with some of the organisms that we study. So why is this list from the WHO so important for some of us? So several years ago, they put out a priority list for bacteria. And that has led to a lot of stimulation in terms of public policy and antimicrobial development. I will tell you that this is behind what we did in our Infectious Disease Society in 2010, where we said 10 by 20, where we wanted 10 new antimicrobial drugs by 2020. And we reached that achievement. But there hasn't been the same kind of approach for fungi, although at the end of the talk I'll show you some new developments that are coming. So the WHO put this together and really focused on the invasive and also neglected organisms. And they tied it into One Health because you can't just think about patients, you have to think about what we're doing across all of our enterprises. We give industrial amounts of antimicrobials to cows and pigs and other animals and birds, but we also spray it on crops. And you can see in this over here that Aspergillus fumigatus in parts of Europe, it's 80% are resistant because of how we're spraying things on our roses and tulips and other plants. So we need to do this in a multifaceted, responsible manner. And as I said, health inequities are really important. What are neglected diseases? What are we not paying attention to that we might be spending tons of money on in London or in Paris or in New York, but we're not spending money on things that are killing people in Africa or in Latin America. So we need to do this again in the holistic approach. And we understand that we're going to see more and more drug resistance because we're using so much of these. So I put a lot of words on here because I talk fast, but I want you to be able to read and see this. So the list is really important because it's supposed to drive interventions. And the bottom here really focuses on what the most important things are: to increase our laboratory capacity, to make investments, not just in research, but in the development of public health interventions, to do it in a holistic manner. And what they did is they looked at how many people die of the organism a year. What's the distribution? Is it only in a very small area, or is it broad? It could be in a small area, but if it's neglected, then we need to pay more attention to it. And you can see the other parts that they looked at as well. Antifungal resistance being very important. So these are the critical high-risk group and medium group. And we're going to pick a couple of different ones from these. And I'm going to go into more detail. And I'm also going to talk about a few other organisms as we go along. So we'll start with Candida auris. So Candida auris, very new. First came up in 2009 in the ear of a person in Japan. In lookbacks of other collections, it turns out that it had been around for a few years before that. And since 2009, we've seen a lot. It's really important because of the high rates of drug resistance that we see and mortality rates. I can tell you in my health system, the mortality rate is only about 15 to 17%, but if you look across large studies, it's about 50% on average across the globe. Not only does it cause disease in us, like Candida albicans, but it can survive in brackish water. It can survive on animals. It can survive on this computer. So it's incredibly resistant and resilient. Because of that, one of the primary areas we see it are in healthcare centers. So if one person has it and someone doesn't wash their hands, or you don't clean the room very well and another person comes in, it spreads across. And it's not just in hospitals, it's in long-term care facilities, and it's a huge problem because it's very hard to get rid of. And I'll tell you a little bit more about that. At the beginning, it was very hard to identify. When you use standard techniques, it was often misidentified as a few other organisms. If you use mass spectrometry, time of flight, you can do a very nice and rapid identification, but not all places on our planet do that. So places that are doing biochemicals still are very much struggling. There are some good PCRs now that you can do to identify it as well. So this was really shocking, and it was really scary. You can see that they stopped this map over a year ago because it's too widespread, so there was no reason to keep updating it. And you can see here a timeline of where it started in Japan and Korea in 2009, and then it, boom, exploded into many different places. And in 2019, they also found another clade, so now there are 5 clades, and that 5th clade was just verified in another study about 2 months ago. So I want to show you the US experience because we have really good data on this, and you can see that in May of 2015, we had a few cases, and then it just went up, and it's continued to stay high. And interestingly, it didn't de novo start in the US. All of our clades that we have there came from other places. So global travel. Early on, it was only in a couple places, and the bright orange is where it was most prevalent, and that's New York and New Jersey where I practice, so we saw a lot of it. And because we started seeing a lot of it, I started working on it. And then it spread. And then it became a requirement that you reported it because it was such an important organism, and we needed to make sure that we weren't sending someone from New York to California as people recover from an illness and not know in California that this person is coming and they need to take precautions there too. And then it spread, and then it spread some more. And this is what we have now. And the black dots are where a year ago there was organisms present and not this year. So it's fairly widespread. This is also the first time that New York was not first. We had as many as about 500 one year of cases. Now we're down to about 250, and California has over 300. So not that many cases, but each one of those cases is very complicated and costs a lot of money to care for. The good thing is overall, Candida auris, as I said, is a small minority of all the species that we're seeing. And for those of you who like to know where it is on a genetic tree, you can see that it actually is very far away from things like Candida albicans and Candida parasilosis. So another troubling thing is that if someone comes in with fevers, it doesn't scream, I've got Candida parasilosis or Candida auris or anything. It's just something you need to work out and figure out. So it's not easy to tell when someone has this or another infection. And it causes almost anything you can think of. Bloodstream, brain, kidney, you name it, it goes there. And with COVID-19, this was one of the infections that we saw increasing, not so much in the US, but in places like India. But also in India, they started seeing a tremendous amount of aspergillus and mucormycosis. And that was probably linked to high doses of steroids that they were giving their patients early on in their infections. In our experience, once someone has this, you can give them gallons of potent antifungals, and the organism still remains on their skin or in their vagina or in their nares, in their nose. It doesn't go away. So that person, when they go to another hospital or they go and visit a sick person, it's very easy to spread it. So it's a huge challenge. And as I said, it also spreads from surfaces. One of the good things is that this is a rare time when both Europe and the CDC are in agreement with how to handle something from a public policy and infection control as well as treatment guidelines. So we are on the same page with how we address this organism in Europe and in the States. In 2016, this was a review. I did not do it, but it showed that there was a lot of resistance, but about 25% were susceptible. And if you looked at different parts of the globe, like East Asia, had high levels of susceptibility. But certain places, like in the US, we had 90% that were resistant to azoles. And in 2019, we started having pan-resistant isolates. And some places have a lot of pan-resistant isolates. What do you do when you have a pan-resistant isolate? You start using combinations, you start using alternative drugs that may have some efficacy, and it doesn't really work very well, and that's why we have the mortality rates that are so high. We know a lot about resistance, so ergosterol, which is a sterol and very important in the membranes, that synthesis can be altered. You can have alterations in what's called a hotspot for regulating other parts of the cell wall structure. And it also changes biofilms. And biofilms are these incredible natural formations of clusters of cells with a dense external polysaccharide mixture as well as other protein components that can be from the host or produced by the organism. So a kind of echinocandin, it's part of the cell wall, beta-glucan that gets dysregulated by this. And you really want to do very careful testing to know that the drug you're giving is going to be effective, and that takes time. It often takes at least 72 hours to know whether something is susceptible or not. So meanwhile, you're giving this medication, you're hoping it's going to be susceptible, but it doesn't always happen. And because central lines, these big IVs are so important, we try and change all of the things that are implanted because it sticks and that biofilm forms. So, what do we know? It's increasing drug resistance. And we know who's at risk, but this group is at risk for so many things that we have to pay attention to things like aspergillus and also gram-negative and gram-positive bacteria also. So it's really hard to know. But because of the high rates of drug resistance, this is the first fungus to be identified as a global threat by the CDC, and it's also viewed that way by European organizations too. So we started working on this because we saw so much of it in my hospital, and this is the first paper that we had come out, and there's a great group of people that I've been able to continue to work with, and I put this slide in because one person was missing from that, but also to show you this slide because it shows you that in order to be successful, it's not a 9-to-5 job, what we do. It's all the time. And sometimes the organism or the mouse experiment needs you to come in on weekends. And so this was a group that was in my lab almost 15 plus years ago, and you can see that they're actually very happy. They did this intentionally to mock me because I was not there. But I also want to highlight that there's an individual here who you may recognize from talking earlier, which is Attila. So Attila was one of my postdocs. And the other thing about science that's so amazing is that it is a family. And you get to continue collaborations, and you're always stronger when you're a community than by yourself. So you're really lucky to have Attila here because he's fabulous. So this group of people, what we did is we did an analysis using a variety of omic techniques to study differences between Candida albicans, this organism that people study all the time, and Candida auris. And you can see here just by the greens and the reds that they're really different. People often lump these two organisms and other Candida species. Oh, it's Candida. No, they're incredibly different. And how they're doing glucose regulation is also really different. And what this really showed us is that Candida auris is much more rapidly adaptable to changes than Candida albicans. So an incredible organism in terms of being able to modify itself. This is Candida parasilosis. It's on the high priority group, and I'm just showing this because Attila and his crew came up with a way of making a rapid evolution, similar to what would happen in us if we have an infection and we're getting weeks of a drug and that organism is still hanging around. So Csaba was also in my lab for a while, came back here, finished his PhD, and is working at the university, and came up with this process of maturing these organisms and showed very dynamic changes in sterols, and also showed that there was a reduction in its virulence. And we did the same thing led by Attila here in Szeged, and he showed by culturing this over time in the presence of these echinocandins that you could get changes in its resistance. And if you changed resistance to one drug, it actually caused resistance to other drugs, including drugs outside of that same category. So jumping to something that impacts the polarization of sterols. And interestingly, he showed that it changed the virulence, but it actually made some more virulent. And usually when you are attenuating something, in order to survive in that new environment, you attenuate virulence, but not in auris, not in some of the strains. They got more aggressive. So really interesting, and something that I think is very exciting for us to continue to work on. So this is an interesting organism because of what we're seeing in our patients, but also it's a story for what we should expect to see more of as our climate continues to change, as we continue to encroach on new environments, as we continue to have global travel. And this was a paper by Arturo Costaval and some friends. Arturo was the person I trained under, and this shows that this organism can be in the wetlands and then travel to farms and then travel to the cities and be in our hospitals. And once it's there, it's not going away. So an important story, and it also links to that One Health, because you can't just look at humans. You have to look at the environment, you have to look at livestock, you have to look at agriculture. And again, we have to pay attention to these things. We're going to hop to histoplasma. So this is an interesting organism. I started working on this because in the height of HIV in New York City, we saw a lot of histoplasmosis because we had people from Puerto Rico or Dominican Republic who came and then got immunosuppressed and had recurrence of this in a very aggressive and deadly manner. But with our capacity to control HIV, we've seen very few of these cases, but I'm still working on it. But interestingly, the range where we see it has expanded. So it used to be just in part of the United States, where the black is over there. But now it's spread to parts of the Northwest, and you can see in that lower picture the hot red spot in Oregon and Washington. People who have never traveled are getting the disease in that area. So it's not from travel, it's not from an organ transplant, it's moving in its environment, as are many other fungi in the United States. So it's also really interesting because it has a high rate of infection, and it's estimated that there are at least 500,000 infections that are new each year, but we only see about 3,000, 4,000 patients in our hospital, and it kills around 200 people. The most I saw one year was around 350. Now that may not seem like a lot, but in the United States, tuberculosis only kills about 400 to 500 people. Malaria only kills about 10 people in the United States. If you look at funding by our government, the amount of money that goes into funding fungi or histoplasma, for example, is like 0.1% of what we're spending on malaria. Now again, as we're sitting here, there's a child dying of malaria in Africa. So the global burden is very large. But again, it gets to where should we spend our money and how do we spend money in equitable manners? I would say we need to spend more money on fungi, and we need to continue to spend a lot of money on malaria and tuberculosis. But there is significant inequalities of where research dollars are being put for some of these very important, complex diseases that we have to deal with. It also is a really important disease in Latin America and in Africa. And in some parts of Latin America, it's one of the most important killers in the setting of HIV. And this paper came out just a couple of months ago showing that in Ghana and Africa, histoplasma was the leading fungal killer in HIV, above Cryptococcus, which historically has been the leading killer in West Africa. So, but again, I'm in New York. Why am I studying this? So it's actually part of pop culture. So there's a Johnny Cash song. Bob Dylan was hospitalized after he developed histoplasma. It's in TV shows like Call the Midwife and Dexter. It's in Spider-Man. But we actually see this in outbreaks, in travelers. People go into a cave and they come back home to New York, and you have 10 kids who went on this trip to Guatemala, all with fevers and new infiltrates in their lungs, and it happens to be histoplasma. So it's a pretty cool organism. And it's really cool because it gets inside of these macrophages and other phagocytic cells, and it's very happy there. Like tuberculosis, it regulates that intracellular milieu where the VATPase is inhibited and other factors are changed. Again, most people are asymptomatic, only about 1% get disseminated disease. And it's been long known that the person who gets the highest inoculum, even if they're immunocompetent, can get very ill. If you're immunocompromised, you also have a much higher risk of getting the disease. So you get these great images. It looks a lot like tuberculosis, even when it heals. So it's very hard to make these diagnoses because the diagnostics are terrible. And you can see some of the other manifestations of disease. So I started in the lab working on this because I was really interested in antibody, and I'd done some really great work in Cryptococcus. Great, not that it was great, but I had a great time doing it. Actually, it was pretty good. But I wanted to know if it could work on an organism that was at least during a majority of its time intracellular. Now, those macrophages that are loaded with cells rupture early on in infection. There are opportunities to have these extracellularly, and you could bind antibody. There had been studies in the 80s and before using polyclonal serum. Polyclonal serum is not great because there's good and there's bad and there's indifferent antibody, and you really need to find the good antibody. And I also want to remind you about what antibody does. And the classic approaches to looking at functions are to look at it as an opsonine or neutralizing virus. And several of these require host factors. So you can opsonize something, but the phagocytosis part is usually very important. So you need to have a neutrophil or a macrophage. And that really drove a lot of the newer insights over the past 15, 20 years, looking at the immunomodulatory aspects of antibodies. And we've looked at changing antigen presentation, clearing antigen, gene alteration. In fact, Attila has some work on that we've never published on histo, so we need to do that one of these days. But and we've found some newer ones too that I don't think I'm talking about today, but tomorrow I think I am. So now we have a whole bunch of different antibodies to the surface of histoplasma, and we've used them for a variety of studies. I want to share this briefly with you, and this is work from Alan, another student of mine who's down in Brazil, and Mariana, who's pictured with their son, was also a student of mine. So not only did they get great work, but they got a great life partner out of being in my lab. But you can see here on the right with the survival graph that giving antibody could protect 90% of the animals with certain of the antibodies. And there was one antibody that had two different isotypes, and one of the isotypes was good in protecting, and the other one was bad. And so we've also looked at why one is good and one is bad in some very interesting studies too. What we found is that these antibodies were profound in terms of how they modulated the host response, in terms of mitigating inflammation, in terms of changing that intracellular interaction of phagosome, cell, and activation of those processes. And we did some other things too, like looking at T-cell activation, and Attila did this when he was in my lab, showing that the protective antibodies significantly changed the activity of processing and presentation of the organism's components. I think it's also really interesting that we think we know everything about an organism, and then you get into the molecular biology, and other people have been looking at this, and they showed that it's not just Histoplasma capsulatum. Now there are several different strains based on genetic analysis, and some of them lack surface moieties and are still fully virulent, and others are just less common because of their geographic location. So they all present in almost the same way, but they have some nuanced differences. So we need to better understand what some of those differences are and why they have so many different species. So the medium priority group, we're going to look at Paracoccidioides, and I bet most of you have never heard of this. Is that true? Anybody? Okay. So what is Paracoccidioides? It's basically in Latin America, and it's one of the most important invasive fungi in that region. It primarily causes pneumonia. In some people, they get a lot of skin manifestations, and you can see here some of the skin manifestations that are prevalent. It has an interesting shape. It often is the middle one, which is a Mickey Mouse form, or what's called the pilot's wheel or captain's wheel on the far side over there, where you see baby cells coming off of the mother cell, so it's like a wheel that's spun around. And it too was thought to be one species, and now with genetics, it's been shown that there are several different species. And again, they all present in a very similar manner, and they're treated in the same way, but they are very distinct in some of their cellular biology. And really interestingly, the primary therapy is sulfa drugs. So sulfa drugs are things like Bactrim, trimethoprim sulfamethoxazole, things that we use for urinary tract infections from E. coli. So it's an antibacterial drug, but it happens to work against this particular fungus. It also works against pneumocystis, another fungus. And you have to treat it for like 2 years or more sometimes. So these are some of the papers that I've published in the past 3 years. So even though it's a very unusual organism, there's a robust community studying it. And one of the things that happens if you're one of a few people studying something is someone sends you an email and says, you know what, you're the worldwide expert on this organism, which is pretty cool. I showed my mom, she was very excited. But you can't always believe this, because if you look at Candida parasilosis, I'm way down there. Attila's almost at the top, but the person above him is one of his students. So here are just two different things that we've done recently. One is a DNA vaccine, and the other is using antibodies, just showing that we're taking various approaches to combat this. And one of the other really interesting things about this is that we're able to advance things into preclinical and possibly even clinical trial much faster because it's a neglected disease and because Brazil has an incredible infrastructure of antibody development and this big pharmaceutical factory in Rio de Janeiro. So this is great, this priority pathogen list, but it's missing some really important things. I'm going to give you just 3 examples. Emergomycosis. It's now one of the most common opportunistic infections in Africa in individuals with HIV. Blastomycosis. It's increasing in the United States, and its geography is spreading in the United States. And then Sporothrix shankii and Sporothrix brasiliensis. There's an outbreak that's going on. I'm going to give you a little bit of the work we've been doing on Sporothrix. So I learned that there's only Sporothrix shankii. I was told over and over there's only one Sporothrix. So it lives on wood and grasses. It's globally present. And it gets injected into the skin through punctures, and it is something that doesn't heal very well. You usually have to give a drug to kill it. And it either spreads through the lymph or sticks exactly where it's been inoculated. And now there are a whole bunch of different Sporothrix species that have been identified. And I want to focus on this brasiliensis, which is only new within the past 10, 12 years. But in reality, they diverged millions of years ago. So this epidemic in Brazil has been incredibly interesting because it's linked to cats, feral cats in the favelas, these poor areas. So it's not the general population who's getting Sporothrix, it's the poor people. And a lot of women who stay home in the favelas and the cats, and they interact and they get this disease. So you can see that in a short time, there were a large number, several hundred, and now there are several hundred every year of these cases. So here is one patient that we worked on. This was a guy who was a construction worker. So in Brazil, if you're a construction worker in the rural parts, you don't have these cranes and stuff that I saw in your city. You're basically putting things on your back and climbing up, to the 5th story with the bricks on your back. So he was a brick carrier. He was very strong. And then he developed this. And you can see here that he's in a wheelchair. And he's got these nodules on his skin. And he's got really bad arthritis. He can't stand. He can't walk. And over 5 years, he had this disease. And he would get treated. He'd get treated for a year. And then he'd be better. And then he'd come back with a relapse. And in the Brazilian hospital, they got serial cultures of him over this 5 years as he's getting more and more antifungals. And we were able to show that as he had this progression of the organism within his body, the Sporothrix got more virulent over time. So it was a boot camp within the person, and that's really scary. And another thing we found was that a woman had 2 different organisms, 2 different Sporothrix. It was easy to figure out because on a plate, some were black and some were white. So the black is melanin. Melanin is something I've spent a lot of my career studying. And it's a factor of virulence. It makes it harder for drugs to kill the organism, and it makes it much more hardy in the environment. And this lady was unfortunate enough to have not just 1 infection with this fungus, but 2. So it's pretty cool. So I've told you a lot of bad things. What's some good stuff? Well, we have some new diagnostics and we have some new drugs. And along with those new drugs, there's really cool work on antibodies and vaccines. And on Wednesday, when I fly back to the States, I'm actually flying back to Los Angeles, where there's a conference in the National Academy of Sciences in the US on another fungus called coccidioides, which is one on the pathogen list too, where they're trying to advance a vaccine for that fungus. So Talaromyces is a really cool organism. When it grows, it puts out this reddish pigment in the media. But it's also neglected. It's the third most common opportunistic infection in Southeast Asia, but most of you probably have never heard of it again. And because of where it is, there's very little science taking place to look at it. It happens to be in the medium priority group for the WHO. So I've been working with a woman in Chiang Mai to try and develop a more rapid laboratory test for this using our antibodies that we've generated to the organism over the years. And you can see here it's like a urine test or a COVID test, and they're pretty simple to make. You just need the right reagents. And so we've made this, and they're using it in Chiang Mai now for diagnostics, and they're trying to send it to the community hospitals to look at too. So hopefully we'll have more information about how good it's working, because we know how well it works on banked samples, and it works very well. Now there's a well-developed immunochromographic test for Cryptococcus, and that's on the critical priority list. And IMI, the company that makes it in the United States, sends it all over the globe, and they've priced it where they thought it was fairly fair. So it's $1 a strip. Well, $1 a strip is too expensive for places in the Philippines or in Thailand. So we made a strip that's only about 30 or 40 cents, and we're using that right now and validating that, and hopefully that one will be sent around with the Talaromyces one. So really important to think not only about what's happening in Szeged and Budapest and Munich, but what's happening in some of these other areas where they don't have resources, and how can we, with our knowledge and the reagents that we've made in our labs, help people across the globe. There was not a new antifungal for almost 20 years. So the echinocandins were the last ones, and that was 2001 when caspofungin was introduced. So in the intervening 20 years, there were some new models of the old drugs, but nothing new. This was the first new drug approved for an invasive mycosis, and actually it was only approved for vulvovaginal candidiasis. So not really a significant invasive disease, but more of a local disease. But once something's approved, you can use it for other things off-label. So this is pretty cool. It works a lot like the echinocandins because it's a glucose synthesis inhibitor, but the susceptibilities are different. The resistance patterns are not the same. So that's pretty cool. And then this total new character here, which is a pyrimidine biosynthesis inhibitor, is in advanced phase 2, phase 3 trials. And I was at a meeting 3 weeks ago where they presented the phase 2 results, and it's pretty cool. It's pretty cool because it works against molds that previously have been very resistant, like Scedosporium, where we often have no drug to use against them. And where it's gotten a lot of uses against coccidioides, especially meningitis from that fungus. And it's been really tremendous to have that activity. And it's been pretty easy to get on compassionate release. So this one hopefully will be released soon. This was the phase track that was put out 2 years ago and is now off of their website. And I'm hoping that they're still slightly on target, but as you understand, COVID really inhibited a lot of the progress of some of these. So it probably won't fully be approved in 2022 because we're nearing the end of 2022. But with the data I saw in the phase 2, and I know where they are in phase 3, it should be approved in 2023. So hopefully that's coming. And in our lab, we've done some development. This is with nitric oxide in a nanoparticle, and it very effectively kills Candida auris and other fungi and other bacteria. Utilizing this would be hard systemically, but again, I told you that one of the most difficult things with Candida auris is getting rid of it from the skin after you've eradicated it from the bloodstream. So this is very easy to apply to the skin. So it's possible that this could be part of an eradication of a patient after they've had that disease. So I know that a few of you are falling asleep. It is hot in here, and I'm talking in English and I'm talking fast. But have we learned anything tonight? And I would say we have, because if you look at this example from 10 years ago, this was an outbreak of a fungus that got into a steroid injection from a pharmacy in Massachusetts. And all of a sudden we had people popping up across the country because it was distributed across the country with this really unusual fungus that we'd seen sometimes in sinus infections, but never in a bone or a joint. And we weren't sure what to do at first because we didn't have susceptibility testing, but we knew a lot about it. And we knew, for example, that melanin was a major product of this. And because we knew some of these things, we knew what drugs should work. And so we empirically started using drugs as we started doing testing to see what really should work, and we were right. So using the knowledge that we have from these other organisms helps us combat the new ones or the surprising ones that change and appear in our patients. So we've learned that there are more and more serious infections from fungi every single year, and that these pathogens are really causing tremendous problems in our healthcare settings, whether it's in Africa with emergomyces or Candida auris in Europe, and Spain in particular has a big problem with it. Latin America, Colombia had a lot. Brazil didn't have a case till a couple of years ago, and now they're seeing a lot more cases. So once it gets in, it doesn't go away. It just gets worse and worse. And as we continue to change our environment and get into our environment, we're going to start seeing more and more of these organisms. And as we keep using drugs in ourselves, in our patients, but also in the environment, we're going to start seeing more and more because we have more and more immunocompromised patients. We do have some new things that I showed you, but there are a lot of unmet needs. So many of the diagnostics that we have that are really great don't work in under-resourced countries, and we need to do better. So this plan by the WHO is really powerful because it touches on all of these. It touches on building infrastructure. It touches on public policy, making people aware and being vigilant. And I think this is something that we all can agree, that it's not just with fungi, but it's with viruses, bacteria, parasites, that we all need to do better and work on what we're doing and translate that to improvements in knowledge so we do better for our environments and for our patients. And I want to acknowledge some of the people that had work that I presented in here, but I especially wanted to thank Attila and his group for all the work that we've done together. And again, to just underscore how that community and partnership is so strong in science and why we all do better. So with that, I will wrap up. And I think I'm going to hang around for beer downstairs afterwards if people don't want to ask a question now, but I'm happy to take questions here too.
Thank you, Josh. And now it's open for questions.
I just wanted to ask how melanin helped fungi be resistant to viruses.
So the question was for the people who are watching online and may not have heard the question, how does melanin protect these fungi? So it's a great question, and many of the melanotic fungi are environmental. So if you are out in the environment and are melanized, you can actually use sunlight and other radiation to translate that into energy. So it's like photosynthesis, and it's real. We published it, and there was a lot of controversy, and then other people reproduced it. It's one of the top papers in PLOS ONE that they've ever had. But it also helps it from the heat and freezing. So it stabilizes the cell wall. It also is like activated charcoal, so it binds toxins. It binds to heavy metals in the environment. It's one of the reasons why fungi are good at remediating toxic areas. It also binds our antifungals. So you have to use higher amounts of antifungals because it's bound in the melanin there. So it works by all sorts of different mechanisms. It is an incredibly interesting structure. And it is the only natural pigment that we don't know what its true structure is. So it's something that I've been studying since 1998, And we just got another grant so I can continue to study it for another 5 years. And I hope I have more answers to that question. But it is an incredible polymer that has a lot of mysteries.
Thank you for your great talk. I am working in the field of antibiotic resistance in bacteria, and I was wondering how is the development of pipeline of new antifungal drugs. So you mentioned that you have a lot of emerging therapies like antibodies, nanoparticles, etc. You have some FDA-approved new antibodies, but what is the pipeline? We are waiting for these very new strategies, or you have something too?
So there are lots of things in development, and the problem is how much it costs to get something from your laboratory into practice. And the manner in which things are being generated from idea to the community has also shifted tremendously. Pharmaceutical companies are getting out of drug development. What they really do now is they wait for you, through government funding, through angel investors, to bring it to a phase 1 or phase 2, and then they come in and they throw money at you and buy it, and then they do it. So the number of people that make it to phase 2 that are really good candidates is maybe 1 in 10. Sometimes in some areas it's more like 1 in 100. There are some novel therapeutics that are very targeted. So phage therapy, which is something which has been incredibly interesting in some of the bacteria. There hasn't been work in fungi really on phage.
Don't you have fungal viruses?
There are some, but there haven't been many that have been identified for Canada, for example. So there have been some large viruses for certain environmental fungi, but not the human pathogenic ones that.
But they don't lyse,
Yeah.
the fungi. So they live together, replicate inside the fungi, but not like in the case of bacteria, they lyse it and they escape for that way. No. The fungal viruses like to be
Yeah.
inside the fungus, and fungus doesn't care at all.
So, but with the phage, one Acinetobacter may be lysed, but another one won't be. So you have to have very specific phage for each bacteria. And mycobacteria also, there's some great work on phage biology too, but those are kind of one-offs. It's not something which is on the shelf for everybody to use. Antibacterials have fallen also. That pipeline and the 2017 charge and the Infectious Disease Society, there is outrage that we don't have newer drugs. Some of the newer drugs that we have are basically taking an old drug and putting a second drug onto it that then makes it work again. And that's great, but it's not novel, it's not new. We replicate very slowly, us humans. A bacteria replicates every 10 or 20 minutes. Candida albicans is one of the fastest fungi, and it replicates every about 2 hours. So other fungi replicate every 8 or 10 hours. So these are all complications, because if you're trying to do drug development and you, with a bacteria, know the next morning if something worked or not, but with a fungus, you have to wait 3 days. Or actually, some of them it doesn't kill, it just changes some morphology, which is enough to then allow the host response to work. So there are so many challenges. That doesn't mean we shouldn't keep trying, and it doesn't mean that we shouldn't work as a community. And there were some great examples that came out of a woman, Leah Cowan's lab in Canada this past year. And both of those papers, using artificial intelligence to identify various compounds, were actualized through collaborations across, one of them I think was 6 different countries, and one of them I think was 4 or 5 countries. This is what we need to do. You can't do it in your own lab, some of these things. You need to have a very active community who have knowledge in various areas. So the work on multiomics in Canada Ars that I did with Attila, we used a friend of mine in Washington State who is at the Pacific Northwest National Labs, which is one of the leading centers for innovations in omic technology and analysis. And he was able to figure things out in a few weeks that would it take? I'd still be working on it now if I tried to do it myself. So you need to use your community wisely. And I was hearing that Attila is putting in a grant with the UK and the US on a really interesting program because each person has unique strengths. And that's what we have to do. We have to find collaborators to drive our information more rapidly forward. It's a long answer.
Other question?
I got a second question from this answer. You mentioned the 3-day replication of some fungi and also the diagnostic problems. So, oh yeah, of course, the patient comes, gets an infection, we hypothesize it as an infection, we give empirical, I guess, antifungal or antibiotic. So do you see in the future coming new techniques like Malditov coming to every clinic to solve this problem, to have a diagnosis in, I don't know, 2 hours to be able to give targeted antibiotic or antifungal therapy? I don't know, what do you have in your department?
So that's a great question. So we use something called Carius, which is one of the platforms that will find 100+ different organisms and does capture some fungi as well. There are a lot of complications with that because sometimes you get an answer and it doesn't make any sense. You know, you get 5 different organisms. They don't have 5 different organisms. Sometimes they could if they had a big abscess in their abdomen. But a lot of times it doesn't make sense. And the data right now shows that depending on who's using this great test, that it has somewhere of a 94% specificity, 94% sensitivity, but only a 50% specificity. Or in some other hands, it was like 80% sensitivity and 40% specificity. So again, if you need that specificity, that's really important. And just knowing that you have the organism doesn't tell you what the susceptibilities are. And there are some techniques where you can find resistance if you know what the organism is, but some of the quantities of DNA are so low that you can't find that out with the samples that you're getting in the Carius. So we don't really use that test except in very specific patient populations. So we'll use it in our complex transplant patients or our advanced left ventricle assist device patients who are waiting to go under heart transplant and have been on antifungals and antibacterials and they're still febrile. So, you know, we'll use them in very targeted cases. And sometimes it's helpful, sometimes it's not. So we're not there yet. And again, some of these diagnostics that are molecular are really challenging because we then miss resistance patterns. So using gonorrhea, it's always great to talk about gonorrhea at a conference. There's significant drug resistance in Neisseria gonorrhea, and we only pick that up when we have people coming back repeatedly to the clinic, and then we finally will do a culture. And so someone can be going for weeks before we know, sometimes months. And that individual may or may not be still having sex with different people, and now you have a community of people with this resistant one, and then someone flew in from Rio and joined the party and then went back to Rio and now they have it in Rio, or they came back to Madrid. And so the lack of doing the cultures, the classical analysis, sometimes causes problems that are unintended. We like how fast the diagnostics are that are molecular, but it sometimes hurts us in the long run. And it's too expensive to do that plus the culture because most of the time you're going to be right just with the diagnostic. So it's a hard question.
And if I may, this is especially true for species like ours, which has this incredible high resistance rate. So even if you know this is a Candida auris infection, there is a high chance that it's pan-resistant. It's like almost 15% now. So if you use, like, what is your first choice in the States? Echinocandins?
Echinocandins.
Yeah. And also for prophylaxis?
It depends on the posaconazole is one of the ones we use a lot of.
So, but even if the first choice is echinocandins, there is a great chance that 50% of the cases it's going to be useless, completely useless. And you know this is our risk. You start the treatment and you fail, right? So that's why I completely agree.
Yeah.
Sometimes the classical way, just do a culture, see the resistant, and tell you for the physician that just forget everything, just give it nistatin or whatever.
Yeah.
So sometimes, or amphor, maybe it helps.
So it's great to have hard things to work on because that means we have a job tomorrow. There's another question over here.
Can you suspect that this person, for example, has Candida auris infection when the galactomannan level is higher in the blood as in other candidates?
So galactomannans are interesting because, again, they're nonspecific. So it doesn't tell you that you have aspergillus versus histoplasma versus Candida auris. And we typically don't follow that level when we're treating a patient with Candida infection because it hasn't been shown to be that helpful. We'll do that with an aspergillus patient and also the arabinomannan. We can follow that too, but the galactomannan has not been a great one to follow. Where I love it the most is with pneumocystis, because with pneumocystis you often get levels of 300, 400, and there are very few things that will do that. So if I have a patient and they come in and they have infiltrate in their chest that looks a little viral, and they have a high beta D-glucan, then it's going to be pneumocystis.
Yeah, and the problem is that Candida stays in the bloodstream for several minutes. It's adhere to the endothelial cells and leave the circulation, going into the deep tissue, and survive mainly in the liver in the case of human patients, and in the kidney in the mouse. But there is very little chance to catch it by this way. Not in the case of Aspergillus, which stays in the lung tissue and producing that galactomannan a lot, and it's released in the blood and it's taken up in the
The flip side there is that with severe Aspergillus infection, the blood culture is positive in less than 15%, often less than 5% in many studies. Whereas with invasive candidiasis, it's about 75 to 80%. But why isn't it better than that? And it's because there are very few organisms in the blood that are enough, though, to cause a very high inflammatory response.
So you mentioned a lot of them are opportunistic pathogens. So if we just boosted our immune system, would it decrease the fungal infections?
So increasing the immune system is something that's really important for certain infections, but not in all. So if you think about what's called IRIS, the immune reactivation inflammatory syndrome in the setting of HIV, we actually do not give antiretrovirals early for some infections. So for Cryptococcus, we wait 5 weeks usually before we give antiretrovirals because we don't want the immune system to come up, because the immune system is a double-edged sword. When you have the white cells there and they start recognizing the fungus, they start barfing out all sorts of toxic components, and you end up getting damage. Not damage to the fungus alone, but damage to host tissue. And so you can kill somebody if you have too robust an inflammatory response. If you look at COVID-19 and when we were giving steroids and who we were giving steroids to, there were times when we were giving steroids to everybody, and that was bad. So the timing of when you do something is really important, and COVID-19 highlights that very well in terms of when and to whom you should be giving steroids. With certain infections, Candida albicans is a great example, where we historically were seeing that in the 80s and 90s, were in cancer patients who had no neutrophils. And so we gave growth factors to get their neutrophils back as fast as possible, because even though we were giving a lot of antifungals, they still were very sick. And once their white cells came back up, it killed the fungus and the patient was okay. With Candida auris, most of those patients have normal white cells, but they had a catheter put in that allowed the organism to get into their blood system. And once it got in, then it forms those biofilms and hides out. And a biofilm basically can protect the cells down deeper. So the antifungal may be killing the top layer of the cells, but it's not necessarily killing the cells close to the endothelial cells. So it's complicated. But yes, we do try and regulate the host. And in those patients that I showed you on all those inhibitors, one of the things we do is we try and stop them. You can't always stop them. Sometimes those are for treating cancer, and so it's balancing what do we do, what do we lighten up on, what do we keep pushing forward on. So the idea of modulating the inflammatory response is incredibly important. We published on the programmed death pathway, and regulating the programmed death pathway in histoplasma was something that completely protected the mice from a lethal infection. Interestingly, our colleague did the same thing in tuberculosis-infected mice, and the tuberculosis took over and destroyed the mice faster than they'd seen in any other model that they had. So balancing the host response, depending on the animal, depending on the infection, depending on the pathway, remarkable differences. So again, you'd really need to know exactly what the organism is to do no harm, to not make the disease worse in that patient. But it's a great idea, and it's something that we actively are working on. In particular, antibodies and administering antibodies to mitigate or modulate an infection has been something that we've spent a lot of time working on. Thank you for that one.
Yeah, I completely agree. This was the same molecule and the same cell. The pathogen was also intracellular, and the fungus was not killing the knockout mice, and the TB was like killing them in one day. That's why we are not millionaires.
Yeah. There are many reasons why we're not millionaires. All right, well, oh, another one?
So, as you mentioned, these fungi take place also in biofilms, and we have drugs for bacteria in biofilms. Do we have any specific drugs for these situations, or we just use the normal specific drugs for the fungi?
So one of the things that's really important is that many of these drugs are really high molecular weight, like 1,000 molecular weight. So if something is that big, it's hard for it to get into these biofilms. There are some compounds like flucytosine, which is a small molecule, and that gets in pretty well, but you can't use it by itself because resistance happens really, really quickly. So you wouldn't necessarily use that for a biofilm alone, or even with amphotericin, because it's probably going to drive resistance. So interestingly, some biofilms, the 1,000 molecular weights work better than the 200 molecular weight azoles. It's something that's been looked at a lot. There's a really cool drug that's in development that was found from a deep sea microbe by our friends in Madison, Wisconsin. David Andy's group is leading that. And that compound does work very effectively in a biofilm situation. So we're hoping to get more information from that. That again is an example of using cross-field activities by linking with a drug discovery person, with a deep sea biologist, with a mycologist. So really cool stuff. All right, we've kept them up past their bedtime, so at least us past our bedtime. Oh my God.
So we can continue maybe.
Sure.